our research. I'm here presenting my research work on novel deep learning-based computational methods for protein cell structure and protein-protein interaction site prediction. I'm currently working as a research assistant in the research lab with Dr. Mohammad Shamsujah Baisar in our department. So let me first give you a brief overview. Genes are translated into proteins. We all know that. In the very beginning, it is basically just a sequence, and it transforms into a particular structure. Let me just take from my pointer. Is my pointer visible? So a sequence that is a sequence of amino acids is transformed. That is, we call it conformation. It's transformed into a structure, particular structure, and then the structure can be used to interact with some other molecules. Those other molecules can be proteins themselves or some other ligands, non-protein ligands. And by such interactions, these proteins take significant roles in our biological functions. Here I have marked the interaction sites of our protein, of this protein, using violet color. So there are a couple of places where we can use deep learning methods or other predictive models. First, we can predict structures from primary sequence, and we're calling this structure prediction. And there are a couple of variants of this, that is secondary structure prediction, tertiary structure prediction, torsion angles prediction. And then using the structure, we can try to predict where these protein are going to interact with some other molecule, and we call it protein interaction site prediction. And then we can also predict the function of this protein from this interaction, or we can predict it from this sequence directly. There's other new type of problem, that is novel protein design. I thought it is very interesting and worth mentioning here. That is, protein sequence can be designed directly from its functions or information of its interaction with other molecules or its structure. So the first research focus in this presentation is the protein cell structure prediction. And most of this information, most of the information in these slides are taken from our paper, SAINT, that is Self-Foundation Augmented Inception Network, Improves Protein Cell Structure Prediction. This paper is published in Bioinformatics. So the motivation behind predicting secondary structure is, as we know that primary sequences are abundant, but we do not have much of the structures, known structures of them. But as we have seen before, the function of the proteins are mostly dependent on their structures, and predicting the structures will be very beneficial using computational methods than experimental methods, which are very costly and time-consuming. Here we have formulated the problem. The problem is that a protein sequence will, each of the letter that represents an amino acid among the 20 types of amino acids is transformed in one of the 3 states or one of the 8 states. There are 2 types of protein secondary structure prediction methods. 3-state prediction is comparatively easy, but 8-state prediction gives us more information about structure. And also from 8 states, we can directly predict the 3 states without using any learnable methods. So we focus on 8-state prediction in this paper. Here are the names of 3 states and 8 states. Here we've shown the history of secondary structure prediction. Xiang et al. Divided the generations of protein secondary structure in 5 parts. In first and second generation, we can see that mostly statistical methods were used to predict the secondary structure. From the 3rd generation, the machine learning methodologies emerged, and recently we have seen the use of hybrid deep learning methods largely and extensively. So what is the primary challenge in this problem? we know that while conforming, each residue may interact with its neighboring residues, that is, the residues that are neighbor in a sequence, or it can interact with a residue that is sequentially farther, but it is near in the three-dimensional structure, that is, after conformation in its three structure. And capturing the information about the long-range interaction and the short-range interaction at the same time using a single method is a very challenging task. So to capture this short-range interaction, previous methods have been using 1D and 2D convolutional neural networks. And for capturing the long-range interactions, these methods have been using RNN-based networks, residual networks, inception-based deeper models, and hybrid networks. In this paper, we proposed a method that is called self-adhesion mechanism to capture the long-range interaction parallelly with the short-range interaction that we captured using 1D convolutional neural networks. The self-adhesion mechanism implemented here has been taken from the paper "Attention is all you need" that has been proposed by Boswani et al. For implementing our method, we have used a data set proposed by Hansen J et al., which has more than 10,000 protein sequences. And for testing our method, we used 5 benchmark data sets. And among these 5, one of them is compiled by us, that is called CASP-FM. For features, we used one of representation, sequence profile, that is PSSM, HMM profile, predicted contact mass that have been predicted by spot contact, and physicochemical properties. For implementation, we've used Keras with TensorFlow backends. Here is the overall architecture of our method. The input features are passed through our proposed 2H3i module. This module here is proposed by us. And after two sequentially placed 2H3i modules, we place a self-adhesion module, and then a conf11 1D convolutional layer, and then another self-adhesion module, and then dense layer. And this pattern is repeated twice, and then the output is passed through softmax normalization. This produces the final output. Our proposed 2H3i module is depicted here. 2H3i represents 2H3i stands for Attention Augmented Inception Inception Network. Here we have augmented the original inception module proposed by Fang et al. By incorporating self-adhesion module into each of these branches. And then these outputs are concatenated to pass to the next module or next layer. Here is the inception module that has been proposed by Fang et al. In their paper. Each of this module or comp layer is basically 1D convolutional layer, which is very useful. It has been proven to be very useful for sequential models, sequential data. Here we have shown our proposed self-adhesion module. The input is passed through 2 parallel layers, parallel paths. In the upper path, it is summed with positional encoding, and then passed through scale dot product attention submodule and multiplied with a learnable parameter alpha. In the lower branch, the raw input is multiplied with 1 minus alpha, and these 2 outputs are summed and passed through batch normalization. The output is passed through the next layer. Here is the detailed overview of the scale.self-adhesion module. The input H is passed through 3 linear layers, producing key, query, and value, very popular concept in self-adhesion mechanism. Key and query are multiplied using matrix multiplication and scaled, and then passed through softmax normalization to generate the attention map, which is then applied on V, that is the value, and this multiplication is the output of this module. Here are the equations for the previously shown module. Here we have shown the accuracy of our module, and we have compared it with previous state-of-the-art methods. Here we can see that Sane outperforms the previous methods in all the 5 test datasets. Here we have also shown the statistical significance of the improvement of our methods, and we have compared it with the previous methods, and we can see that in 4 out of 5 test sets, the improvement is statistically significant. Here we have also shown the F1 score comparison. Here we can see that out of the 8 labels, in 6 labels, Saint outperformed the previous methods. As we can see that our model generates some attention map, and we know that attention map gives some interpretability in the deep learning methods. And it is very important to have interpretability in a deep learning model that has been used for some biological tasks. So we have done some interpretability study, and we have found that there is a pattern in the attention metrics that correlates with the actual physical phenomena. That is, for any single residue, the subsequent residues in a sequence, in a protein sequence, primary sequence, the distance should be increasing. But after a certain threshold, it starts decreasing, and after some time, it also again starts increasing. That means here we can see that for D, the distance is increasing, and then near P, it is the maximum, and then it starts decreasing again. That means K becomes closer to D than P. And then again, after K, it gets further and further. It shows a possible bend in the protein structure. And we have plotted this along with our attention matrix. That is, the background is basically our attention matrix that has been produced by our model. And here, our model produces a high attention along this certain residue, that is K. And the same pattern is repeated for W here. And we have seen this pattern to repeat for 93.32% of the proteins of our largest test set, that is test 2016, that's been proposed by Hansen et al. That's been generated by them. Sorry. For further analysis and discussion, we refer to our paper, Saint. Now we go to our second research focus, that is protein-protein interaction sites prediction. Most of the information of this slide has been taken from our paper, age-aggregated graph addition networks and transfer learning for protein-protein interaction sites prediction, which is currently under review at ISMA 2021. So why is protein-protein interaction sites prediction so important? Proteins, the functions of proteins are mostly defined by their interactions with other proteins. And in often cases, the partner of the protein with which it is interacting is not known, and we don't know any details about the partner. So it is important to know the interaction sites of a single protein. And proteins tend to interact with any other protein in a particular way. That is, it must interact with the other protein using some certain residues. So we focus on predicting those residues. So why using graph neural networks? Previous state-of-the-art methods have been using sequential models. That is, they have represented proteins as sequences, but these representations misses out of very important information. That is the structural information of the protein that is available in the B2B database. Graph neural networks are very good to capture structural information in data if the data can be represented as a graph. So we took on the challenge to represent any protein as a graph and then apply graph neural networks on them. Here is how our proposed pipeline works. Here we have shown a protein, simplified protein. And here the blue sections are basically interaction sites, known interaction sites. We represent this protein as a graph. And in this graph, we can see that the nodes are basically the residues, and the edges are computed using k-nearest neighbor graphs. The details are explained in the later slides. We then represent each of the
ছোট্ট একটু ইন্টারাপ্ট করি রেসিডিউটা কি জিনিস একটু এক্সপ্লেইন করবা
রেসিডিউ হচ্ছে কি প্রোটিনের অ্যামিনো অ্যাসিড রেসিডিউ
আমার আসলে কোনো ধারণা নাই অ্যামিনো অ্যাসিড রেসিডিউ কি জিনিস যদি তুমি আমাকে একটু ব্যাকগ্রাউন্ড দিতে তাহলে ফলো করতে সুবিধা হতো
তাহলে একটু আমার ইনিশিয়াল একটা স্লাইড আচ্ছা ফর এক্সাম্পল আমরা এই প্রোটিন সিকোয়েন্সটা দেখতে পারি প্রতিটা জিন হচ্ছে গিয়ে একটা পসিবল একটা অ্যামিনো অ্যাসিডে কনভার্ট হয় তো প্রতিটা ট্রিপলেট আর কি একটা করে পসিবল অ্যামিনো অ্যাসিড কনভার্ট হয় এরপরে 20 পসিবল অ্যামিনো অ্যাসিড আছে তো এই অ্যামিনো অ্যাসিডগুলোর একটা সার্টেইন কি বলবো কম্বিনেশন একটা সার্টেইন স্ট্রাকচার সবসময় জেনারেট করে যেটাকে আমরা বলি হচ্ছে গিয়ে এক্সপ্রেশন লাইক জিন থেকে এক্সপ্রেস হওয়ার সময় এটা একটা ইন্টারমিডিয়েট স্টেজ আমরা বলছি আমি যদি এখানে আমার ওই রেসিডিউর পার্ট নিয়ে কিছু বলা নাই আমি সরি এক্সট্রিমলি সরি
can I add
yes sure
রেসিডিউ হচ্ছে প্রোটিন সিকোয়েন্সে যে ক্যারেক্টারগুলো আছে এক একটা ক্যারেক্টার এক একটা রেসিডিউ
মানে এক একটা অ্যামিনো অ্যাসিডই একটা রেসিডিউ
যে কোন মানে এটা সিকোয়েন্সের মধ্যে একটা ক্যারেক্টারকে একটা রেসিডিউ বলে আর কি এটা এখন প্রোটিন সিকোয়েন্স স্ট্রাকচার কমিউনিটি এটাকে রেসিডিউ বলে
আচ্ছা ওকে আমি আসলে এর সাথে পরিচিত ছিলাম না বলে জানি না
প্রেসিপিটেটেড রেসিডিউর কোনো সম্পর্ক নাই
আচ্ছা ঠিক আছে থ্যাংক ইউ
ওই অ্যামিনো অ্যাসিডটা পেপটাইড বন্ড তৈরি করার পরে একটা করে ওয়াটার মলিকিউল ছেড়ে দেওয়ার পরে যতটুকু থাকে ওটাকে রেসিডিউ বলতেছে এই কারণে নামটা হচ্ছে রেসিডিউ মানে যতটুকু রেসিডিউ অবস্থায় আছে
আচ্ছা থ্যাংক ইউ ভাই তুমি কন্টিনিউ করো প্লিজ
So I think I was here. Yeah, so each of the residue, amino acid residue, and for each of the ages, we generate some feature representation, and then along with the feature representations, we pass our graph representation of the protein through our proposed method, that is eGrid. And eGrid generates the very putative interaction sites. That is, which of these residues are interacting with other protein molecules. Here we can see that the green parts are the interaction sites that's predicted by our model and true, that is true positive, and red is false negative, that is it should have been an interaction site, it should have been predicted as an interaction site, but it is not, as we can see. And here we have predicted some false positive. And if we are given the true interaction sites, we can compute the error and pass it as a back propagation to our learnable module, that is eGrid. eGrid contains some learnable parameters. And during inference, only the protein is given and we don't know the interaction sites. And so we just predict this protein with protein interaction sites. So as I have already told, our protein is represented as a graph that contains the structural information. Each node is an amino acid and each age is connected, each residue is connected with its nearest 20 residues. So we use k-nearest neighbor graph to compute the ages for each of the residues. In our final method, k is equal to 20. For generating node-level features, we used a method called ProBERT that has been proposed by Alnagar et al. For each of the residue, it generates a feature representation or embedding Instead of PSSM HMM, we use this feature representation, and as we have seen, for more than 100 proteins, it generates feature represented within a couple of seconds. Whereas with the same computer, if I try to generate PSSM, that's my computer. If I want to generate PSSM for every single protein with average length, it takes more than 2 hours. So it is very time efficient. Now we have to check whether it gives as good prediction as PSSM does. We'll see in the later slides. For age features, we're using 2 values. That is, first one is node-to-node distance, and then the second one is relative orientation of the residues that are connected by an edge. Our architecture is described here briefly. Our architecture has 3 parts. The first one is local feature structure, which is an 1D convolutional layer. And the second one is our proposed graph aggregated, age-aggregated graph addition layer. And the third one is our node level classifier. As we have seen, each of the residue can be an interaction site or not, so our prediction is node level. Here we have shown the whole architecture for a small protein with 13 residues, or 13 amino acids. And here lambda is the local feature structure, which is basically a 1D convolutional neural network with window size 3. So within lambda, only 3 residues can fit. And with the initial feature representation of each of the residues, we can compute the new representation of any residue based on the information of the other residues that are within this window. That is, for generating the new feature representation of 2, we will not consider only the information of 2, but also 1 and 3, that is sequentially neighbor. So now what we are left with is the information of its structure. And for this, we use our proposed age-aggregated graph addition layer, which we represented as epsilon. Here we can see that 10 is not near to 2 in its sequential in its sequence, but it is near in its 3D structure. So we generate a graph. It has 4 nodes in the graph, and the center node is 2. We want to generate a new feature representation of 2 based on the information of its neighbors. That is the residues that are connected to incoming residues, with incoming edges. And then we generate a new representation of 2 and pass it through our linear classifier. So here we have explained our age-aggregated graph addition layer. Here, 2, 1, 3, that is the same as shown above. Here, this trapezoid module aggregates the information of the nodes as well as the edge and passes through an aggregation or summation layer. And for each of the edges, it is done, and the aggregated information is concatenated with the previous information of the center node. This generates the new feature representation of the center node. And this procedure is applied on each of these residues at the same time. We have shown only for 2 here. So the new feature representation of 2 is passed through a linear layer and then a sigmoid activation function to generate a propensity value of the residue to be an interaction site. Here we have expanded this trapezoid module. Here the node features hj and h2. J represents the neighbor. This can be 1, 2, 1, 3, or 10. This neighbor's information and the node's information are linearly transformed and concatenated with the linearly transformed version of the H feature. And this concatenated version is then again linearly transformed and passed through a nonlinearity, and then a softmax normalization to generate an attention score. This attention score is normalized for all the neighbors so that we get a probability distribution over the neighbors. This shows us which neighbor has more importance for this center node to be predicted as an interaction site. This opens up new doors for studying interpretability of this model. This attention square is then multiplied with the feature representation of the neighbor and then the age information that is linearly transformed. And this summation is then passed through. And this output is shown here in using this black line, as I've mentioned before. Here we have shown the summary of our dataset. This dataset has more than 400 proteins, and among them 70 are used for test and 352 are used for training. Here we can see that it's a very unbalanced dataset, and this dataset has been compiled by Sazan Agile and is currently widely used for this problem sets. We have shown here a comparative analysis of our model with our previous methods. Here we have, we can see that our method is performing better than all the previous state-of-the-art methods in almost all the metrics, except for recall. And in recall, GAT PPI outperforms our methods. But interestingly, GAT PPI is also proposed by us in our research work. And GAT PPI is in the second place for the rest of the modules, for the rest of the metrics. Sorry. Here we have shown the interaction size of the protein. Here we can see that the second best method, that is Delphi, has a lot of false positive interaction sites, and this is the true interaction size rate of the protein, and here is our predicted one. Here we have also shown the long local contact. Here we have also shown the accuracy of our methods and its comparison with the previous methods. When the number of local contacts or the protein length increases, here we can see that, as expected, when the number of local contacts or protein increases, all the methods perform worse. But what we have found that the improvement of our method gets more and more significant when the non-local contacts and the protein lengths increase. Here we have shown that how ProteBERT performs as a feature extractor. Here we have used DPPSP features that includes PSM and HMM, as we have mentioned before, and ProteBERT uses none of this. We have seen that using ProteBERT, our methods perform significantly better than using PSM-based features. This proves that transform learning using ProteBERT-based features actually increases the performance of these models. Also, as our model generates attention scores, here is a chance of studying interpretability, which we did, and we have found that there is a significant correlation between the source node's propensity of interaction and the destination nodes. And this correlation coefficient is 0.782, and it has p-value of 0, which shows it is very much statistically significant. We have also studied the interpretability of the ages and its attention scores. What we have seen that if our method predicts a residue to be an interaction site, the neighboring residues probability of being an interaction site highly correlates with their corresponding attention scores, and it is also statistically very much significant. Here we have showed the interpretability of our method. This residue 90 has been predicted by our model as an interaction site, and as I have told before, higher attention score correlates with its neighboring sites to be predicted as interaction sites, which we can see here.
Can you go back a few slides?
Yes, sure.
slide 39. Yeah, slide 39. Okay. So, so in the curve in A, so for the red curve, you see there is an uptick towards the end. So can you explain why this is happening?
You mean this one? Well, these two, both of these models are proposed by us, and actually there isn't
that's not the problem. So
yes,
the why the
so,
AUPRC is increasing in the last two steps for the red curve. For the green and blue, I see the trend is downwards.
okay, I think this isn't enough. There isn't enough data to explain why for exactly this one it goes up, because there are only 7 values for this analysis, and for the last one it goes up. Maybe if we had more data, we may be able to see that it goes down again. So,
yeah, I would suggest like get some more data points after this, and then see what the exact trend looks like. Okay, so and then go to slide 41, 42. Yeah, what test did you do here? P-value 0 means nothing. So never report a p-value 0 in a paper. What statistical test did you do exactly here?
So I did correlation test using Spearman correlation coefficient.
So is it a Pearson correlation coefficient or Spearman or Kendall rank correlation? Okay. So p-value 0 means there is something wrong with the test.
সাজান এটা পুরো 0 ছিল না, খুব ছোট ছিল আর কি, রাউন্ড করে 0 আসছে হয়তো, রাইট?
জি স্যার,
হ্যাঁ সেটাই, এটা 0 ছিল না, এটা খুব ছোট ভ্যালু, কিন্তু এটা 0 না।
যখন এই p-value প্রেজেন্ট করো তখন এমন ভাবে প্রেজেন্ট যে p-value is still less than 10 to the minus 16, something like that. 0
এটাকে রাউন্ড করতে না কখনো,
কখনোই ভালোভাবে দেখা হয় না।
হ্যাঁ, মানে এটা আসলে লেখার সময় সেভাবেই লেখা আছে যে সাবস্টেন্সিয়ালি লেস দেন 0.05, যেটা হচ্ছে থ্রেশহোল্ড, কিন্তু এখানে হয়তো স্লাইডে ভুলে 0 লেখা আছে।
হ্যাঁ এটাই।
Okay, go ahead.
So here we have shown the runtime analysis. We have compared our method with previous state-of-the-art method DPP-ISP, and here we can see that our model is significantly faster than these methods for the longest as well as the shortest protein in our datasets. We refer to our paper Buet for further analysis and discussion. For future work, we want to focus on backbone torsion angle prediction using SAINT and contact map prediction. And using the architecture of Buet, we want to predict the pairwise protein interaction size prediction. That is, given 2 protein structures, we want to predict where these 2 proteins are about to interact. And for also, we want to explore DNA binding sites of a protein and a ligand, that is non-protein binding sites of a protein. Thank you everyone. This is a great asking question.
আমার কয়েকটা কোশ্চেন ছিল একটু এর আগে, মানে এখানকার শুরুর দিকে স্লাইড একটু, যখন হচ্ছে প্রোটিন ফিচার, সরি নোড ফিচার আর এজ ফিচার নিয়ে যখন বলা হচ্ছিল, না পরের হ্যাঁ
এজ ফিচার,
হ্যাঁ নোড ফিচার, হ্যাঁ তো এই মানে প্রথম হচ্ছে নোড ফিচার আর এজ ফিচার, মানে এগুলি কোন ধরনের, মানে ফিচারটা কি রিপ্রেজেন্ট করে, রেসিডিউর ফিচারগুলো আর এজের ফিচারগুলো?
ডিপ লার্নিং এ আমরা যেটা করি যে প্রতিটা টোকেনকে একটা, যেমন সিকোয়েন্সিয়াল মডেলে আমরা প্রতিটা টোকেনের একটা রিপ্রেজেন্টেশন তৈরি করার চেষ্টা করি। আর ওয়ার্ড টু গ্রাফের জন্য ওয়ার্ড টু ভেক্টরের মতো অনেকটা কিছু, আচ্ছা হ্যাঁ ওয়ার্ড টু ভেক্টরের মতো অনেকটা, ওইখানে হচ্ছে আমরা সিকোয়েন্সের প্রতিটা টোকেনের জন্য চেষ্টা করি, আর এখানে হচ্ছে কি গ্রাফের প্রতিটা টোকেনের বা নোডের জন্য চেষ্টা করি।
মানে ওটা কি কোনো ধরো মানে সরি, ওটা কি কোনো ধরেন ইয়া আছে, মানে কোনো ইন্টারপ্রিটেশন আছে যে ধরেন এই রেসিডিউর আমি এই ফিচার ভেক্টর পেলাম, তো এটার এরকম কোনো কিছু কি আছে, মানে এখানে যে ফিচারটা ইউজ করা হয়েছে
এই ফিচারের যেটা প্রোটবার্ট, জি, প্রোটবার্টের একটা ডাউনসাইড হচ্ছে কি এই ফিচারের এক্সপ্যান্ডেবিলিটি এখনো খুব একটা ক্লিয়ার না। ফর এক্সাম্পল বার্টের আমরা যে ল্যাঙ্গুয়েজ মডেল ইউজ করি ন্যাচারাল ল্যাঙ্গুয়েজ প্রসেসিং এর জন্য, সেখানে বার্টের যে এমবেডিংটা ঠিক সেরকমই এমবেডিং। এটা বেসিক্যালি একটা বার্ট যেটা ট্রেন করা হয়েছে পুরা ইউনিকোড ডেটা সেটের উপরে। তো যেমন এখন বার্টোলজি নিয়ে স্টাডি হচ্ছে অনেক, যে ওই এমবেডিংটা আসলে কি মিন করে, কি রিপ্রেজেন্ট করে, এখন পর্যন্ত খুব ক্লিয়ারলি জানা যায়নি। তবে কয়েকটা কেসে দেখা গেছে যে ওই এমবেডিংটা কন্টাক্ট ম্যাপের সাথে কোরিলেট করে, কয়েক কেসে দেখা গেছে সেটা হচ্ছে কি ডিরেক্ট ইন্টারাকশন সাইটের সাথে কোরিলেট করে।
আচ্ছা, আর এজ ফিচারটাও এখানে খালি এই দুইটা ফিচার, মানে নোড টু নোড ডিসটেন্স আর রিলেটিভ ওরিয়েন্টেশন, এই দুইটা নাম্বার রাখছি আর কি, রাখা হয়েছে নাকি?
জি 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 জি, এটা টোটালি এক্সপ্যান্ডেবল। এটা
এই ডিসটেন্সটা কি মানে ফিজিক্যাল ডিসটেন্স নাকি?
জি জি, ফিজিক্যাল ডিসটেন্স। প্রোটিনের স্ট্রাকচারে দুইটা অ্যামিনো অ্যাসিডের সবগুলো অ্যাটমের মধ্যে ডিসটেন্স নিয়ে সেটা অ্যাভারেজ করা হয়েছে।
আচ্ছা আচ্ছা আচ্ছা, আরেকটা কোশ্চেন ছিল হচ্ছে এর আগে যে অ্যাটেনশন, এর আগের যে এটা ছিল এই 2H যেটা ছিল,
জি জি,
আর্কিটেকচার যেটা ছিল, তো এই যে 2A3I এটা তো মানে এই মডেলটা ইউজ করা হয়েছে ইয়ার জন্য, প্রোটিনের সেকেন্ডারি স্ট্রাকচার প্রেডিকশনের জন্য, যে আমি একটা সিকোয়েন্স দিব, সে আমাকে 8টার মধ্যে যেমন একটা ক্লাস বলে দিবে ওটা। সো এখন এটার মানে যে 2A3I মডিউল যেটা, সেটার পিছনে মোটিভেশনটা কি ছিল ডিজাইনের পিছনে? মানে খুবই দেখলাম অনেক ভালো কাজ করে, এন্ড হচ্ছে মানে নোবেল একটা আর্কিটেকচার, তো এটার পিছনে মোটিভেশনটা কি ছিল এভাবে সাজানোর?
জি আচ্ছা, তো বেসিক্যালি এই এতটুকু আর্কিটেকচার হচ্ছে কি ফ্যাং এটালের প্রপোজ করা। তো আমরা এই আর্কিটেকচারটা ওদের পেপার থেকে নিয়েছি, কারণ ওরা রিসেপশন মডিউল যেটা হচ্ছে গুগলের প্রডিউস করা, গুগল নেট একটা খুব পপুলার ভিশনের জন্য ইউজ হওয়া একটা আর্কিটেকচার, ওরা সেটাকে মডিফাই করে সেকেন্ড স্টেজের প্রেডিকশনের জন্য। এবং ওরা হচ্ছে গিয়ে খুব ভালো এভলিউশন স্টাডি দেখায় যে এরকম সার্টেন আর্কিটেকচার ওদের এই প্রেডিকশনের জন্য অনেক ভালো কাজ করে। আমরা সেটাকে অগমেন্ট করি হচ্ছে গিয়ে জি 1 2 4 এই প্যাটার্নে যদি রাখা হয়।
আচ্ছা,
তো আমরা সেটাকে হচ্ছে গিয়ে অগমেন্ট করি যে হ্যালো কি
না হ্যাঁ ঠিক আছে, মানে এটার পিছনে কোনো কি কারণ ছিল যে এটা ভালো কাজ করে কেন, বা এরকম?
অফকোর্স এটা কেন ভালো কাজ করে সেটার অবশ্যই এক্সপ্লেনেশন আছে, আমি যদি এখানে যাই। সো বেসিক্যালি এটা যে একটা সিকোয়েন্সিয়াল মডেল, সো কতগুলো টোকেন সিকোয়েন্সিয়ালি আছে। তো এখানে কোন একটা এটার উইন্ডো হচ্ছে গিয়ে 1, এটা 3, এটা হচ্ছে গিয়ে হ্যাঁ 3। তো এটা যেটা ক্যাপচার করবে সেটা হচ্ছে গিয়ে পরপর দুইটা টোকেনের পর একটা টোকেনের ইনফরমেশন। দ্যাট মিনস এটা আসলে জাস্ট ওই টোকেনটার ইনফরমেশনটাকে একটা লিনিয়ার ট্রান্সফরমেশন দেয়, এটা আসলে এর বাইরে কিছু করে না। কিন্তু এটা যেটা করে সেটা হচ্ছে গিয়ে পরপর 3টা টোকেন এর মধ্যে কোনো ধরনের রিলেশন থাকলে সেটা ক্যাপচার করে। দ্যাট মিনস যদি কোনো ধরনের শর্ট ট্রি ইন্টারাকশন থাকে সেটা ক্যাপচার করবে এইটা। কিন্তু আমি যদি এটাকে পরপর সিকোয়েন্সিয়ালি দুইটা প্লেস করি, দেখানো যায় যে 3 আর 2 5, 5টার জন্য 5টার মধ্যে ইনফরমেশন থাকলে সেটা ক্যাপচার করে, 5টার মধ্যে কোনো ধরনের ইন্টারাকশন থাকলে। তারপর যদি আমি আরেকটা প্লেস করি তাহলে 7টা। দ্যাট মিনস এই প্যাথ দিয়ে যদি একটা সিকোয়েন্সের ইনফরমেশন পাস হয়, তাহলে ওই সিকোয়েন্সের যে কোনো রেসিডিউ তার আশপাশের 7টা রেসিডিউর সাথে যদি কোনো ধরনের ইন্টারাকশন করে থাকে সেই ইনফরমেশনটা এনকোডেড হয়ে পাস হবে। তো এটা গেল জাস্ট ইনসেপশন মডিউল। এখন আমি যদি এই আর্কিটেকচারটা আবার লাইক রিকার্সিভ ওয়েতে আরেকবার করি, দ্যাট মিনস এই প্রতিটাই হচ্ছে গিয়ে এই পরের স্লাইডে দেখানো এই মডিউলটা প্রতিটা, এবং সেই জিনিসটা যদি আমি সেম ওয়েতে আরেকবার রিপিট করি রিকার্সিভলি, তাহলে দেখা যায় যে ইনফরমেশন আরো বেটার ক্যাপচার করতে পারে শর্ট ইন্টারাকশন।
আচ্ছা আচ্ছা, শর্ট লেন এন্ড মানে 4টা সিরিজগুলোর মধ্যে আসলে অনেক লং রেঞ্জও করতে পারতেছে এরকম কিছু,
অনেক লং রেঞ্জ, বাট এটা একটা লিমিটেশন আছে যেটা আমরা ক্যাপচার করার জন্য সেলফারেশন মডিউল নিয়ে আসি। যেমন এটা আমি যদি লং রেঞ্জের লিমিটটা মানে না রাখতে চাই, সেই কেসে কর্নফ্লেয়ার দিয়ে কোনোভাবেই পসিবল না। কেন? কারণ হচ্ছে গিয়ে কর্নফ্লেয়ার সবসময় হচ্ছে গিয়ে একটা উইন্ডোর বাইরে দেখতে পারে না। তাহলে যদি সিকোয়েন্সিয়ালি আমি অনেকগুলো কনফ বা ইনসেপশন মডিউল রাখি, তখন হচ্ছে কি আগের একটার থেকে এনকোড ইনফরমেশনের বাইরে হয়তো উইন্ডো সাইজ ডিভাইডেড বাই 2 এই পরিমাণ ইনফরমেশন সে আরো অ্যাগ্রিগেট করতে পারে ডিপেন্ডিং অন দ্য স্ট্রাইট। তো এরকম করে করে হলেও আমার হচ্ছে কি যদি আনডিফাইন্ড একটা প্রিভিয়াসলি আনডিফাইন্ড একটা প্রোটিন সিকোয়েন্সকে আমার ইনপুট করতে হয়, তাহলে আমার ইনফিনিট সংখ্যক কনফ্লেয়ার পরপর বসাতে হবে। আমি শিওর না বুঝতে পারছি কিনা।
না
মানে
মোটামুটি
একটা সার্টেন নাম্বার অফ কনফ্লেয়ার যদি আমি পাশাপাশি বসে, সেটা যে আর্কিটেকচারই বসানো হয়, সেটা ইনসেপশন হোক বা শুধু ইনসেপশন মডিউল হোক, সেটা কোনো সময় হচ্ছে গিয়ে একটা মানে বিরাট লেন্থের বা যে কোনো লেন্থের প্রোটিনকে ইন্টার করতে পারবে না। যত বড় হবে সে একসময় সে ব্রেক করবে। কনফ্লেয়ার এটাই হচ্ছে মানে সিকোয়েন্সিয়াল মডেল ইম্পোর্ট করার সময় ল্যাকিংস।
এটা বেশ ভালোই লাগছে, সুন্দর লাগছে এটা। আরেকটা আমি আসলে এটা জানি না, সেটা হচ্ছে প্রোটিনে যখন হচ্ছে প্রেডিক্ট মানে প্রোটিন ইন্টারঅ্যাকশন সাইট প্রেডিক্ট করতেছিল, তো সেখানে কি আসলে ইন্টারঅ্যাকশন সাইটটা কি পার রেসিডিউ আমার কি করতে হয়, প্রেডিক্ট করতে হয় এরকম কিছু?
জি মানে ইন্টারঅ্যাকশন সাইট, আমরা সাইট বলতে আর কি একটা করে রেসিডিউকে মিন করছি, সারফেস রেসিডিউ। তো আমি যদি প্রতিটা রেসিডিউ
ইন্টারঅ্যাকশন সাইটটা কি কোন একটা মানে এরিয়ার মতো হয় না নাকি? আমি আসলে জানি না নাকি সেটা।
তো এইখানে এটা এক ধরনের রিপ্রেজেন্টেশন আছে, সেটা হচ্ছে গিয়ে সারফেস রিপ্রেজেন্টেশন। মানে এটা আমরা যেমন গ্রাফ রিপ্রেজেন্টেশন করেছি, সেটা না করে আমরা যদি 3D সারফেস দিয়ে রিপ্রেজেন্ট করতাম, পয়েন্ট ক্লাউড দিয়ে বা এরকম কোনো একটা মেথড দিয়ে, তখন সেই কেসে আমি হচ্ছে গিয়ে পুরো পয়েন্ট ধরে ধরে জিওমেট্রিক ওয়েতে মানে প্রেডিক্ট করতে পারতাম। বাট যেহেতু সারফেসের দেখা যায় যে কতগুলো রেসিডিউ ওই যে সারফেসটা আমার ইন্টারঅ্যাক্ট করবে, সেই সারফেসের আন্ডারে সার্টেন কিছু রেসিডিউই পড়বে। তো আমি যদি ওই রেসিডিউগুলোকে প্রেডিক্ট করতে পারি, তাহলে আমার খুব হাই অ্যাকুরেসিতে আমি ইন্টারঅ্যাকশন সাইট প্রেডিক্ট করতে পারি। তো আমরা একটা সাইটকে এখানে একটা রেসিডিউ হিসেবে মানে বলতে পারি, সারফেস রেসিডিউ হিসেবে। আপনি যেটা বলছেন সেইটাও একটা মানে আরেকটা ওয়ে, এবং এই ওয়েতেও স্টাডি হয় জিওমেট্রি ডিপ লার্নিং দিয়ে।
আচ্ছা থ্যাংক ইউ, এটাই কোশ্চেন ছিল এগুলো।
আর কোনো কোশ্চেন থাকলে আমি I'll be happy to answer.
আচ্ছা, একটু অফ ট্র্যাক একটা কোশ্চেন করি। যে গুগলের একটা পেপার মাঝখানে আসলো, বেশ হইচই হলো, ওটা কি দেখছিলা? ওই পেপারে
AlphaFold 2
আচ্ছা
AlphaFold স্যার দেখেছি, কিন্তু AlphaFold 2 টার পেপার স্যার এখনো আসেনি স্যার। 2 টা হচ্ছে হইচই ফেলে দিয়েছে হচ্ছে স্যার, অনেক বেশি ভালো অ্যাকুরেটলি হচ্ছে গিয়ে স্ট্রাকচার প্রেডিক্ট করতে পারছে আর কি, অলমোস্ট 90 ওদের সেই 90% অ্যাকুরেসি।
আচ্ছা, সেটার পেপার এখন আসে নাই
স্যার, কিন্তু AlphaFold 1 এর থেকে ওদের জাস্ট মানে যেটা যতটুকু জানা গেছে যে ওখানে জাস্ট স্যার ট্রান্সফর্মার বেসড বা বার্ড বেসড ল্যাঙ্গুয়েজ মডেলিংটা ওরা এক্সট্রা করেছে। আর AlphaFold 1 এর যে মেথডটা সেটাও খুব ইন্টারেস্টিং ছিল স্যার, এবং সেটা দিয়েও ওরা আসলে গতবার যে কাস্ট 13 সেটা তো ওরা স্যার AlphaFold 1ই হচ্ছে গিয়ে হাইয়েস্ট স্কোর করেছিল, কিন্তু AlphaFold 2 হচ্ছে গিয়ে সেটাকেও বিট করে অনেক দূর চলে গেছে।
ওকে। তবে মানে ওই কেউ কেউ একটু স্কেপটিক্যাল, কারণ গুগল আবার এর আগেও কিছু জিনিস নিয়ে যতটা দেখা গেল শুরুতে হইচই, অতটা না। দেখা যাক আর কি এটা কি হয়।
হ্যাঁ দেখা যাচ্ছে।
So today I am going to present my work, which is a global alignment-free approach to phylogeny estimation from genome schemes. My name is Bristy, and I have conducted this work with Ahnaf Faisal and Metin Balaban, and this work is supervised by Dr. Mohammad Samsudjiha Baiji and Dr. Siavash Mirara, and this is a collaboration with University of California, San Diego. So before beginning the presentation, let's first talk about the organization of this presentation. First, I will show some definitions in the introduction section. Next, I will talk a little bit about the necessary backgrounds that will help understand this work better. And then I'll jump to the methods and pipelines, followed by experimentations, future directions, and our contribution. So our main goal here is to estimate phylogenetic trees. So what are exactly phylogenetic trees? Here we are seeing a phylogenetic tree of the vertebrates. This tree shows the evolutionary relationship among different species or entities. So why do we need phylogenetic trees? As the true evolutionary histories cannot be known, phylogenetic trees can be very good hypothesis of what has happened in the past. So from where we are going to estimate phylogenetic trees in the study, we will be using genome schemes, and from them, we are eventually going to estimate phylogenetic trees. So let's first talk about genome and the genome schemes. Genome, it consists of all the genetic material of an organism. It consists of DNA or RNA in the RNA viruses, and a genome scheme is the fragments of DNA. It contains the information about the high copy fraction of the genome. Before going to the problem definition, let's first look at another definition, that is reverse complement of a genome. Each nucleotide in a double-stranded DNA molecule is paired with its Watson-Crick counterpart. For example, A's counterpart is T and C's counterpart is G. So let's look at an example. Let's suppose the original sequence is ACGTA. The reverse sequence will be ATGCA. And the complement sequence will be TGCAT. So combining reverse and the complement, the reverse complement will be TACGT for this original sequence. So let's go to the problem definition. Given a set of genome schemes, we have to estimate the phylogenetic tree of a respective taxa set. So let's jump to the background of this work. We are using alignment-free methodology. So let's first look what alignments are. Sequence alignment is a way of arranging DNA, RNA, or protein sequences to identify regions of similarity. After aligning the sequences, distances can be calculated. But here we are not doing it. So what are the advantages of not doing it? Firstly, sequence alignment is a very costly operation. So avoiding these steps makes the pipeline faster. Secondly, accurate identification of structural similarity does not always guarantee shared ancestry of the residues. And thirdly, multiple sequence alignment introduces many bias and errors at many levels. So avoiding these steps can make the work more accurate. So how will we get the distances in an alignment-free method? We are using k-mers in this case. So let's look at the k-mer-based methods. What is a k-mer? In bioinformatics, k-mers are subsequences of length k contained within biological sequences. So our work is solely based on previous work done by Dr. Siavash Mirara, that is k-mer. K-mer is also an alignment-free tool for estimating the distance between two genomes. So k-mer treats the genome schemes as simply as low coverage bag of reads. Firstly, it uses k-mer profiles to estimate amount of sequence error and coverage, and then it uses the hashing technique of MASH to compute checkered. We will go to these definitions in the later sections. So what MASH does is MASH extends the min hash dimensionality reduction technique, and MASH uses k-mer sketches. Sketch means we have to use an exact number of k-mers instead of all the k-mers. So using smaller number of k-mers, the original sequences distances can be estimated very rapidly with bounded error. But MASH uses a heuristic technique. If we want to use a more accurate technique, we have to compute all the k-mers, and Jellyfish does that trick. Jellyfish gives us all the k-mers of a sequence, and it can give k-mers from k equals to 1 to k equals to 31. After getting the k-mers, we use substitution models to estimate distances. So what a substitution model is, the substitution models are Markov models that describe evolutionary changes over time. This substitution model gives the evolutionary distances between pair of sequences. After getting these evolutionary distances between all pair of sequences, we use various distance methods such as neighbor joining to get the phylogenetic tree. So the simplest substitution model is JC69. This model assumes equal base frequency for all the bases, that is 1/4 for A, T, C, and G, and equal mutation rates. The only parameter that this model uses is alpha, that is the overall substitution rate. So this picture shows the JC69 model, and we can see that the substitution rate from one base to another is always alpha in this case. This is the most simplest model. For more general case, we can use TK4 model that assumes different substitution rates for the 4 bases among all the bases. So the rates of base substitution used in TK4 model are alpha, beta, gamma, delta, and epsilon. That is shown in this figure. You can see that the figure is almost the similar as JC69 model, but this is a more complex model as there are 5 parameters here. And so after getting the distances, we use various distance methods to accurately estimate phylogenies. We have so now we have distances between genome schemes. Our goal is to get the phylogenetic tree. We use FastME tool to infer phylogenies. It is mainly based on neighbor joining methods. So let's jump to our methods and pipelines. In this section, first we will describe our distance estimation technique by using a set similarity measure, and then we will describe our pipeline. The main idea is to, for our alignment-free distance estimation method, is to decompose sequences into k-mers. So we are assuming the sequences as the outcome of a random process which copies a genome and introduces mutation at each position with a fixed probability D. If the length of the sequence is L, then there are total number of L minus K plus 1 k-mers in the genome. The probability that a k-mer is identical in both the genomes is 1 minus D to the power K. I'm not going to the details of this because of the scope of this presentation. So we are using Jaccard index for the similarity measure between 2 k-mer sets. It is known as intersection over union. Suppose we have 2 sets of k-mer, A and B. The Jaccard index will be A intersection B over A union B. The relation between Jaccard index and genomic distance is this. However, to get phylogenetic trees, we need phylogenetic distance between the 2 genomes, not genomic distance. So genomic distance differ from phylogenetic distance due to variable rates of mutation, insertion, deletion, duplication loss, or repetitions. There are several Markov models to infer phylogenetic distances in terms of genomic distances. We have seen in the previous section 2 methods, JC69 and TK4. JC69 is the simplest method. If we have genomic distance D, then JC69 distance will be minus 3/4 ln 1 minus 4/3 D. And if you want to use more complex models, then we can use TK4 model. So let's see how TK4 model estimates these distances. We can see that there are total 16 combination there can be total 16 combination at each site. P, Q, R, and S are the frequencies of their occurrences in 2 sequences. For example, S stands for the probability that the 2 sites are equal. P is the probability of transition type differences, and Q and R are the probability of transversion type differences. So the genomic distance in this case will be the sum of P, Q, and R, which is shown here. So thus TK4 model in our method, we can use genomic distance like this. For computing the P, Q, R, and S described in the previous slide, we devised a replacement technique for the 4 bases. For each I and J belongs to ATCG, we replace I with J, and we denote genomic distance for this replacement is Dij. So let's suppose we want to replace A with C. So if we replace A with C in here, then we can see that these two will be equal, and this 2R2 distance we won't be getting anymore. So DSC will be the sum of 2P1, 2P2, 2Q1, 2Q2, and 2R1. So D minus DSC will give us R. Similarly, we can get all the other parameters of TK4 model, P, Q, and S by different type of replacements. Finally, we use the following formula that is described in TK4 paper to compute the phylogenetic distance using those P, Q, R, and S values. So now let's look at our pipeline. First, we find the Jaccard index of all pair of genomes. Finding the then we have to find the distance of from J, and then we replace one base with another for finding the TK4 parameters P, Q, R, and S. In this step 4, we have to find phylogenetic distance for all pair of genomes using the TK4 model. And finally, we will build the phylogenetic tree using BioNG method of FASTMA. So let's look at the experimental settings. We have used 2 methods for estimating the Jackards. First, for the first one, we used MASH to determine the Jackards. MASH uses mean hash algorithm to hash only S number of k-mers of a large sequence. In the following slide, we will be briefly describing MASH's mean hash algorithm technique. Suppose we have 2 sets A and B of k-mers. A is the set of k-mer of the first genome, and B is the set of k-mer of the second genome. MASH first hashes these k-mers and finds the union of these hashes and keeps the lowest S number of k-mers in the database. For this case, we have A is equal to 5. So you can see that the bigger values that were 128, 139, or 104, 127 that are not in the in union set. And finally, we find the intersection using these 3 sets. So this is the MASH's mean hash algorithm. We can see that was a heuristic algorithm. For more accurate estimation, we are using Jellyfish. Jellyfish tool provides all the k-mers of the sequence as Jellyfish provides a large number of k-mers. We are encoding these bases with 2 bits instead of 8 bits. So we are encoding A with 11, C, G, and T with 10, 01, and 00 respectively. Thus, we represent a k-mer with a binary string and save the corresponding integer in our k-mer database. For example, if we have a k-mer like this, our encoding will be this, and we are saving the integer representation 813 in our database. So for replacements, we read the integer representation and then convert it to bit strings. For each base, we put the first bit of one set, first bit of the whole string to one set, and the second bit to another set. Thus, we divide the whole bit string into 2 sets. Then we do set operations to convert one base to another. This will be more clear with an example. Suppose we have to replace A with C. So our k-mer is ATCAG, and after replacing, it will be CTCCG. So the initial encoding is this. If we have to replace one A with C, we have to replace 11 with 10. So we are putting these red bits into set 1 and these black bits into set 2. If we do these bitwise operation on set 2 will look like this. And after combining set 1 and set 2 again, the replaced encoding will look like this. You can see that in the first base, it was an A, which was an A11 here that has been replaced with 10, similarly with this case. So we do this for all type of replacements and build our k-mer database. So finally, we save all the encoding of all the k-mers to Python sets and find the intersection and union of them and compute jackets. We do the same thing for all pair of taxa and 12 type of replacements. Now let's talk a little bit about our data sets. We have 4 data sets: the Drosophila dataset, yeast dataset, Anopheles dataset, and Bombus dataset. Apart from this, we have used Bombus and Drosophila datasets for simulations. In case of simulations, we have used SecGen tool for simulating these genomes. Let's see the results. The first graph here shows the sketch size selection for MASH. In the Drosophila dataset, we can see that S is consistent, sketch size S is consistent from 10 to the power 5 to 10 to the power 8 region. And in the yeast dataset, we can see that this is consistent from 10 to the power 5 to larger. So we are using 10 to the power 5 in all our experiments. And this is a graph of k-mer selection. We can see that for sketch size greater than 10 to the power 5, the graph is mostly consistent. So we can use any value of k from these values. We are using k equals to 31 in our cases. Finally, we constructed the phylogenies of real Anopheles dataset and simulated Bombus dataset as the final step of our pipeline. For real biological Anopheles dataset, both k-mer and our method with MASH produces the same tree, which is shown in the next slide. So we can see that this is a reference tree, and this is the k-mer tree. Our tree with MASH is the same as this tree with just one branch mismatch. So for the Bombus dataset, we ran 8 simulations. We used k-mer produced tree as the backbone tree of all the simulations for Bombus dataset. We ran our method with MASH and k-mer again on the prepared simulated datasets. So this table shows the comparison of our method with MASH and k-mer. So we can see that branch mismatches with k-mer is 0 for all the cases, and our method with MASH still shows some mismatches. But the good thing is that when the distances are lower, our method with MASH gives 0 error. So now we will talk about the running time. We ran our method with MASH and compared the results to k-mer. For the Bombus datasets, k-mer takes roughly 10 minutes to compute the whole pipeline, whereas our method with MASH takes roughly 120 minutes. The method with jellyfish is still in progress due to time constraints. The whole jellyfish pipeline takes around 10 days for 19 taxon Bombus dataset, even after doing all the encodings. So let's talk about the discussion and future aspects of this work. We have introduced a new method of phylogenetic estimation. Ours is a traditional distance-based method with an interesting twist. Instead of computing phylogenetic distances immediately from the genomic distance, our method leverages the assumptions of TK4 model and computes accurate phylogenetic distance. However, our method with MASH did not perform well. So currently, our concern is to focus on the running time of the jellyfish and get more accurate estimates of phylogenetic distances. Thank you. If you have any questions, please feel free to ask them.
Can you go to slide 50?
Sure.
Yeah, so here, did you only consider odd k-mer sizes?
Yes, we have considered only odd k-mer sizes.
Is there any particular reason for that?
No, there is no particular reason. Okay. Instead of doing all the tests from 21 to 31, we have only done 6.
And this choice of 21 to 31 seems rather arbitrary. People usually check for k-mer sizes